In this new segment on Diencephalon, let us begin learning about thalamus. Diencephalon is made up of two major parts, a pars dorsalis which includes thalamus and epithalamus, and pars ventralis which includes hypothalamus and subthalamus. Pars dorsalis and pars ventralis are separated from each other by a shallow hypothalamic sulcus. Thalamus is by and large the largest part of the diencephalon. Each thalamus is an ovoid mass having four surfaces: a medial surface, a lateral surface, a superior surface, and an inferior surface. The two thalami are connected with each other through interthalamic adhesion. Anterior end of the thalamus is narrower, known as thalamic tubercle. Posterior end of the thalamus is broader, it is known as pulvinar. It shows two oval swellings on the surface known as medial and lateral geniculate bodies. Anteroposteriorly, the thalamus measures about 4 centimeters. Its breadth and height are about 1.5 centimeters each. Long axis of thalamus makes an angle of about 30 degrees with the midline. That is because the two thalamic tubercles, or the anterior ends of the thalami, are converging towards each other. On the lateral aspect of the thalamus, there is a band of grey matter known as reticular nucleus, which is separated from the rest of the mass of the thalamus by means of a sheet of white matter known as external medullary lamina. Now coming to the relations of thalamus, medial surface of the thalamus is related to the cavity of third ventricle. Lateral surface, that is outside the external medullary lamina and reticular nucleus, is related to posterior limb of the internal capsule. This posterior limb of the internal capsule is located between thalamus and lentiform nucleus. Superior surface is related to the cavity of lateral ventricle, that is the central part of the lateral ventricle. The superior surface is actually covered by a sheet of white matter known as stratum zonale. Between the stratum zonale and the cavity of lateral ventricle, there are a series of structures from medial to lateral end, which include the choroid plexus of the third ventricle, stria medullaris thalami, body of fornix, choroid plexus of the lateral ventricle, stria terminalis, thalamostriate vein, and body of the caudate nucleus. So all these structures will separate the superior surface of the thalamus from the cavity of lateral ventricle. Inferior surface of the thalamus is related to hypothalamus and subthalamus. The upper surface of the thalamus as well as anterior end of the thalamus is related to fornix. Now between the fornix and the anterior end of the thalamus, anterior end otherwise known as thalamic tubercle, between the fornix and this thalamic tubercle, we have what is known as interventricular foramen, or otherwise known as foramen of Monro, which communicates the cavity of lateral ventricle with the cavity of third ventricle. So anterior end of the thalamus is related to interventricular foramen. Posterior end, or the pulvinar end, is related about to the pineal gland, pineal stalk with the pineal recess. Within the pineal stalk, we have habenular commissure above and posterior commissure below. All these structures together form what is known as epithalamus. The pulvinar end is related below to the superior colliculus of midbrain. Now extending from the interventricular foramen, running backwards and downwards, there is a shallow sulcus known as hypothalamic sulcus. Now this will continue at its posterior end with the cerebral aqueduct within the midbrain. Now this hypothalamic sulcus is the one which separates pars ventralis from the pars dorsalis. Now pars ventralis is basically made up of the hypothalamus, which is below that. Pars dorsalis is made up of thalamus and epithalamus. So we are seeing the thalamus above and the parts of the epithalamus which are located in and around the pineal gland and its stalk. On the surface of the thalamus, we are also seeing the cut section of interthalamic adhesion. Now coming to the nuclei of thalamus, Thalamus is an ovoid mass which is divided by a Y-shaped band of white matter known as internal medullary lamina into 3 groups of nuclei: anterior nuclear group, medial nuclear group, and the lateral nuclear group. We are seeing the same thalamus from the side profile here, again divided by the internal medullary lamina into anterior group, medial group, and the lateral group. Now, lateral group is further divided into multiple subnuclei. So let us find out what they are. First, it is divided into a dorsal tier and a ventral tier. The dorsal tier is further subdivided into 3 nuclear groups. From front to back, they are lateral dorsal, lateral posterior, and the posterior most portion known as pulvinar. On the posterior surface of the pulvinar, we are seeing 2 oval swellings which form the medial and lateral geniculate bodies. Previously, this medial and lateral geniculate bodies together formed what was known as metathalamus, yet another part of the diencephalon, but now these 2 parts are considered as integral parts of the thalamus itself. Now, coming to the ventral tier, again that is further subdivided into 3 main nuclei: ventral anterior, ventral lateral, and ventral posterior nucleus. Ventral lateral nucleus has again 2 subparts, an anterior pars oralis and posterior part caudalis. Similarly, ventral posterior nucleus is divided into 2 parts: ventral posterolateral and ventral posteromedial nuclei. Now, apart from these 4 groups, that is anterior group, medial group, dorsal tier of the lateral group, and ventral tier of the lateral group, we also have reticular nucleus on the lateral aspect, intralaminar nucleus within the internal medullary lamina, and midline nucleus which is located along the medial surface as well as extending into the interthalamic adhesion. So these are the various nuclear groups within the thalamus. So let us find out the details about individual nuclei. Now, anterior nucleus receives its input from the mammillary body via the mammillothalamic tract, and it sends its output to the cingulate gyrus because anterior nucleus of the thalamus is a part of Papez circuit. It is involved in regulation of alertness and attention, as well as being part of the Papez circuit, it is involved in acquisition of memory. So any lesion to the anterior nucleus will result in deficits in memory, wherein the patient tries to fill up the gaps in his memory through confabulation or making up his own stories. That's known as Koroskov's syndrome. Now, coming to the medial group of nuclei, which is made up of mainly one large nucleus known as mediodorsal nucleus or MD nucleus. It has 2 subparts, a magnocellular part and a parvocellular part. Magnocellular part receives input from the piriform cortex. Parvocellular part receives input from the prefrontal cortex. If you remember, olfactory sensation is the only sensation which does not relay in the thalamus before reaching the cortex. So the primary olfactory cortex is the piriform cortex, where the olfactory information has already reached. Now it will project to the thalamus, wherein it will be reaching the magnocellular part of the medial group of nuclei. The outputs from the medial nucleus will be the magnocellular part will project to posterior orbitofrontal cortex. Now this is the olfactory association area, where the olfactory sensation will be discriminated as to identify what is the type of the smell. The parvocellular part, of course, will be having reciprocal connection with the prefrontal cortex. So the magnocellular part is involved as a part of the olfactory pathway. Parvocellular part, of course, has a performs higher functions very similar to prefrontal cortex because of its connection with the prefrontal cortex. So any lesion to the medial group will result in decreased anxiety, aggression, as well as amnesia. The features will be very similar to those seen after prefrontal lobotomy. Now coming to the ventral tier nuclei. Ventral anterior nucleus first. Again, it has 2 subparts, a principal part and a magnocellular part, but both of these parts will be receiving inputs from globus pallidus internum, premotor cortex, substantia nigra pars reticulata, and frontal eye field. All of these are motor related centers. And its major output is also to the motor areas in the frontal lobe as well as to the anterior parietal cortex. So this ventral anterior nucleus influences motor activities on the contralateral half of the body. Now coming to the ventral lateral nucleus, it has two parts, an anterior pars oralis and a posterior pars caudalis. Anterior pars oralis receives input mainly from globus pallidus internum, that is part of the basal ganglion, and posterior pars caudalis will receive input from the contralateral deep cerebellar nuclei. Outputs from the anterior part is to the supplementary motor area and premotor area. So it is this portion, that is anterior part, is receiving input from the basal ganglia and projecting to supplementary motor area and the premotor area. So this part is involved in planning the motor activities. The posterior pars caudalis receives input from the deep intracerebellar nuclei and it will project straight to the primary motor cortex or area number 4. So that is involved in initiating activities or motor activities on the contralateral half of the body. So this ventral lateral nucleus is active in both passive as well as active movements in the contralateral half of body. In case of Parkinsonism, we find what is known as essential tremors or resting tremors. When these tremors become exaggerated, they become debilitating. So certain parts of CNS are ablated through surgery, that is stereotaxic surgery, wherein we ablate certain parts within the central nervous system to reduce these tremors. One of the area used for that ablation, stereotaxic ablation surgery, was ventral lateral nucleus. However, currently, for reducing the tremors, the more preferred sites will be globus pallidus internum and subthalamic nucleus. Now coming to the ventral posterior nucleus. Now this is the nucleus which is sending in or receiving the general sensations as well as taste sensations. General sensations, which include touch, pressure, temperature, pain sensations from the contralateral half of the body, they will reach via the medial lemniscus and spinal lemniscus, and they relay in the ventral posterior lateral nucleus. Now general sensations coming from the contralateral head region will arrive through either medial lemniscus or trigeminal lemniscus, and taste sensation is arriving through solitariothalamic tract. Now these fibers will be relaying in ventral posterior medial nucleus. From here, the projection to the cortex is to the primary somatosensory cortex, and taste sensation fibers will be relaying to area 43, which is the lowermost part in the postcentral gyrus itself. So this nucleus, the ventral posterior nucleus, will act as a chief sensory relay station for both general senses as well as for taste sensation. Now coming to the dorsal tier, both lateral dorsal and lateral posterior nuclei will receive inputs from pretectum and superior colliculus in the midbrain, as well as from the cingulate cortex, parahippocampal gyrus and hippocampal formation of the limbic lobe. And outputs will be to the cingulate cortex as well as to parahippocampal gyrus and parietal cortex. Now coming to the pulvinar, which is the posteriormost part in the dorsal tier, it receives inputs from the superior colliculus as well as from the retina through the optic tract, and its output is to visual areas, specifically the parastriate area, and also to the prefrontal cortex. Its connection from the retina to the visual area, that is involved in what is known as blind sight. So even when there is actual blindness, the person can feel as though the light is moving, that is the blind sight, that is mediated through the connection to the pulvinar. And through its connection to the prefrontal cortex, it helps in modulating perception, cognition, memory, and also probably in modulating pain. Now behind the pulvinar, we have a pair of geniculate bodies. The medial geniculate body, which is further subdivided into 3 nuclei, medial, ventral and dorsal nuclei, that is actually part of the auditory pathway. So it receives its input from the inferior colliculi and also from the superior colliculi via the inferior brachium. Among the 3 nuclei, the ventral nucleus within the medial geniculate body has got tonotopic arrangement, that is the sound waves which are low pitched are represented laterally, and high pitched sound waves are represented medially. And from the medial geniculate body, they project to the primary auditory cortex through the sublentiform part of the internal capsule. The ventral nucleus will project directly to the primary auditory cortex. The other two nuclei, that is medial and dorsal nuclei, they will project to other auditory areas as well as to insula. So this medial geniculate body is a part of auditory pathway. Similarly, the lateral geniculate body is a part of the visual pathway. Now, instead of being divided into nuclei, this lateral geniculate body shows 6 laminae. It will be receiving its input from the optic tract. Now, each optic tract will be carrying fibers from both retinae. It will be carrying fibers or the information from the ipsilateral temporal retina and contralateral nasal retina. So within the lateral geniculate body, the contralateral nasal retina fibers will relay in laminae 1, 4 and 6. Ipsilateral temporal retina fibers will relay in laminae 2, 3 and 5. Among these 6 laminae, lamina 1 and 2 are made up of magnocellular laminae, and they will receive input from Y type of ganglion cells which are fast conducting and rapid adapting. The remaining 4 laminae, that is 3, 4, 5 and 6, they are parvocellular laminae. They receive input from the X type of ganglion cells which are actually slow conducting and having a sustained response. In addition, there is a third type of ganglion cells known as W cells. These are even responsive to light. They will relay in the interlaminar zones as well as in the lamina S, which is located just above the lamina 1. Within this lateral geniculate body, there is visuotopic arrangement. That is, the fibers coming from the periphery of the retinae, they are located anteriorly, and the fibers which are coming towards from the vertical meridian, that is closer to the macula, they are located posteriorly. From the lateral geniculate body, the output is to primary visual cortex or area number 17, or otherwise known as striate cortex. So lateral geniculate body is a part of visual pathway. Now coming to the intralaminar nuclei, they are divided into 2 groups, an anterior group and a posterior group. The names of the nuclei are mentioned here, but among all these names, just remember the largest of the nuclei belonging to the posterior group known as centromedian nucleus. Anterior group will be receiving inputs from reticular formation, superior colliculus, pretectal nuclei, prefrontal cortex and various areas from the parietal, temporal and occipital cortices. Whereas the posterior group nuclei will be receiving inputs from almost all motor areas, that is globus pallidus internum, substantia nigra pars reticulata, deep cerebellar nuclei, pedunculopontine nucleus, primary motor area, premotor area and supplementary motor area. Their output again is to similar cortical areas, that is anterior group will be projecting to prefrontal cortex and various cortical areas in the parietal, temporal and occipital cortices, whereas posterior group will be projecting to mainly the motor areas. So the function of intralaminar nuclei, it helps in mediating cortical activation because of its input from the reticular formation. The posterior group is also involved in sensory motor integration. Any lesion to the intralaminar nucleus will result in thalamic neglect, that is, both corporeal and extracorporeal contralateral half will be neglected if the intralaminar nucleus will be involved in one side. It may be also associated with motor neglect, that is, the contralateral half of the body will show reduced movement. If there is bilateral damage to the intralaminar nuclei, that will result in akinetic mutism, similar to what we get after the lesion to the supplementary motor area. Now coming to the midline nuclei, they receive inputs from the hypothalamus, periaqueductal gray and reticular formation, and their output is to hippocampal formation, amygdala, nucleus accumbens, cingulate cortex and orbitofrontal cortex. So because of their connections, you can make out that these are actually part of the limbic structures, and they have role in memory and arousal. Lesion to that would also result in seizure activity, so maybe they have a role in regulating seizure activity. Now coming to the reticular nucleus, which is the last group of nucleus. Now when we have seen connections of all the other nuclei within the thalamus, we find that all the nuclei within the thalamus connect to different parts of the cerebral cortex. Similarly, all these nuclei will receive input from different parts of the cerebral cortex. So all these corticothalamic fibers and the thalamocortical fibers will send collateral fibers to the reticular nuclei. So basically they are informing the reticular nuclei before they send output to the cortex, or cortical fibers before they reach thalamus, they are again sending information to the reticular nuclei. Within the reticular nuclei, there are specified areas which are visual region, somatic region and auditory region. So corresponding functional fibers will be relaying in those regions. And main output from the reticular nucleus is to the main body of the thalamus itself. So what is its function? It helps in gating the information through the thalamus, what is known as saliency. So cortex will be receiving a flood of information via the thalamus. It could be from multiple modalities: visual input, auditory input, somatosensory input, gustatory input. So the reticular nuclei will pick up what is important, which need to be sent first in the background of the noise. So that is what is done by the reticular nucleus. Now all these nuclei can be further classified as relay nuclei. Now these are the nuclei which are receiving specific input, that is either visual or auditory or somatosensory input, and they project to discrete cortical areas like visual areas or auditory areas or primary somatosensory cortex. So they belong to relay nuclei. They are the specific nuclei. So the nuclei which are included in this group include all the nuclei in the ventral tier, medial geniculate body and lateral geniculate body. So all of them have specific functions. They receive input from a specific area and they project to specific area in the cerebral cortex. The second group of nuclei include association nuclei. Now these are basically projecting to cortical association areas. So barring the nuclei in the ventral tier and the medial and lateral geniculate bodies, all other nuclei can be grouped under association nucleus. And then there is a third group known as non-specific nuclei. They are part of general arousal system. They receive information from the reticular formation and they will project to almost all the areas within the cortex. So the reticular nuclei and the intralaminar nuclei, they belong to this non-specific group of nucleus. This is one way of classifying the nuclei in the thalamus. The output from the thalamus to the cerebral cortex is through 4 thalamic radiations. Each of these thalamic radiation will pass through a specific part of the internal capsule. These fibers begin from a particular set of nuclei within the thalamus and they have a specific destination within the cortex. So let us find out what they are. The anterior thalamic radiation passes through the anterior limb of the internal capsule. These fibers begin from anterior and medial nucleus of thalamus and reach the frontal cortex. The posterior thalamic radiation passes through the retrolentiform part of the internal capsule. The fibers begin from lateral geniculate body and they reach visual cortex, that is the area number 17. Superior thalamic radiation will be passing through genu as well as posterior part of the posterior limb of the internal capsule. These fibers will begin from ventral tier nuclei, that is ventral anterior, ventral lateral and ventral posterior nuclei. They will be reaching primary motor cortex, premotor area and primary somatosensory cortex. Inferior thalamic radiation will be passing through the sublentiform part of the internal capsule. These fibers begin from medial geniculate body and they will be reaching auditory cortex. So these are the 4 thalamic radiations. Now coming to the functions of thalamus, it acts as a sensory relay station, acting as a gateway to cortex for all the sensory modalities except for olfaction. The nuclei which are involved in the sensory relay activity are the VPL nucleus, that is ventral posterolateral nucleus, ventral posteromedial nucleus, medial geniculate body, and lateral geniculate body. The second function is it influences motor activities. The nuclei involved in this function are the ventral anterior, ventral lateral, as well as the posterior part of the intralaminar group of nuclei. It has a role in regulating mood, behavior, and memory by its connection to the limbic lobe centers. So the nuclei involved are the anterior nucleus and the medial dorsal nucleus. It also has a role in regulating consciousness, sleep, and alertness. The nuclei involved in this function include reticular nuclei and the intralaminar nuclei. Now coming to the blood supply of thalamus, the superior part of the thalamus is supplied by choroidal branches of posterior choroidal artery, which is a branch of posterior cerebral artery. The inferior part of the thalamus is supplied by thalamoperforate branches coming from the posterior communicating artery. And the posterior part of the thalamus is supplied by thalamo-geniculate branches, which are given from the posterior cerebral artery. Apart from the various clinical significance which we have described when we were dealing with individual nucleus, there is one more condition known as thalamic syndrome, otherwise known as Dejerin-Roussy syndrome. Now this is basically, it has clinical symptoms of hyperpathia, hyperalgesia, and allodynia. Hyperpathia is heightened sensations for any sensory input. Hyperalgesia is exaggerated perception of pain. And allodynia is even any other modality of sensation is perceived as pain, like a simple touch with a cotton wool is perceived as painful stimulation. So all these form features of thalamic syndrome, otherwise known as Dejerin-Roussy syndrome. The cause is not known. It could be thalamic stroke, but not necessarily all thalamic strokes will present with thalamic syndrome features. But the features are unmistakable. Whenever thalamus is involved, the patient will present with hyperpathia, hyperalgesia, and allodynia. So thank you very much. Hope you enjoyed this video. You can visit this site for other neuroanatomy videos.